Oh, there it goes. All right. All right, let's go ahead and get started for today then. Hopefully everybody got everything turned in over the last week and over the weekend. Remember, the best way to do well in this class is to keep up with doing the problems at the back of the chapters. Work on that as you go through the chapter, do the problems, write down where you stumble on a problem. You know, I understand all the way up until this point, but here's where I'm having trouble. And then come see me or the ISL for help. This is where we left off last time, so we're finishing up chapter 5 today. And another way in which inheritance of genetic information happens, in which you get phenotypes that don't completely look like they should, is in this way. So everything we've talked about in the chapter up until now, the inheritance of the genetic material has been exactly the same. It's just interactions of the alleles lead to phenotype ratios that are different than what you would expect. Cytoplasmic inheritance is different because it's not nuclear inheritance. So the chromosomes are all in the nucleus. So what the heck is in the cytoplasm? Well, it might surprise you that mitochondria and chloroplasts have their own DNA. So these are former endosymbiotic microbes that learned to play nice with early versions of eukaryotic cells, and we decided to keep them and they decided to keep us. So mitochondria are very good in harvesting energy, and chloroplasts are very good, well, mitochondria are very good in harvesting energy from sugars, using cellular respiration, conducting electron transport, and chloroplasts are very good in harvesting energy from the sun and turning it into chemical energy in the form of sugars that both plants and everything else uses as energy. If it were not for chloroplasts, we would not exist. We don't make our own energy. Every plant or animal protein or sugar you eat comes from energy that was derived from the sun, just about. So there are some microbes that can live on chemical energy alone, but they're not a major food source for any animal that I know of, at least not terrestrial animals. So mitochondria and chloroplasts were former prokaryotic organisms. Therefore, their genome is not linear chromosomes like ours, but circular DNA molecules. They have only one DNA molecule. This is bad in that anytime you get a mutation, you can't be heterozygous for it because there is only one allele. So mitochondria and chloroplasts, again, originally endosymbionts, but because they've been with us for so long and we provide them lots of nice goods in exchange for their services, they have lost a lot of the genes that are required for living on their own. If you pull mitochondria and chloroplasts out of the cell, they just die because they're specialized and therefore they've lost the genes they don't need to live. Right? So these guys are found in multiple copies in the cytoplasm. They replicate as the cell is growing, and so when a cell goes to divide, roughly double the number of chloroplasts and or mitochondria, well, and mitochondria and or chloroplasts are present. So all organisms have to have mitochondria. Plants have both mitochondria and chloroplasts, so everybody needs to have mitochondria. All right. So interestingly, in most species, mitochondria and chloroplasts are inherited entirely from the female. In humans, that makes sense. In mammals, that makes sense. So the mitochondria in sperm are actually, I'm not going to draw it on the iPad because it's not worth it for this, but here's the sperm head is different shaped in different organisms. So the sperm head contains the DNA and enzymes necessary to penetrate the coat around the egg. And then right here between the head and the tail is a little part of the sperm called the midpiece. All the mitochondria are right here because this is where the energy needed for working the flagellum is located. So most sperm, Drosophila are an exception, when they meet the egg, the head will fuse with the membrane of the oocyte and dump just the genetic material in, so the chloroplasts don't go in at all. Other sperm, like those of everybody's new favorite model organism, C. elegans, it is everybody's new favorite model organism, I'm hoping. Don't tell me if it's not, you'll hurt my feelings. Surprisingly, their sperm don't look like that. They look like this. They're amoeboid. So they have a cell body and a pseudopod, and they crawl around. So when these guys fuse with the egg plasma membrane, everything from the cytoplasm goes into the egg, including the mitochondria. But the egg has a plan, immediately massacres all the male-derived mitochondria, so even when they get in, the inheritance of mitochondria come only from the female, and plants chloroplasts as well. So a demonstration of that can be done by looking at what phenotypes you get in the progeny and comparing that to the phenotypes of the parents. So here are some cells. These are either mitochondria or chloroplasts, let's just say mitochondria. And the red and the blue are not what color the mitochondria normally are. Actually, these are mitochondria. See the little membranes in there? So they're different alleles. So let's say that the red allele is some kind of a mutation that's not going to be good. A mutation has happened somewhere, but there's both kinds here. And so although cells can't be heterozygous because these are not on chromosomes, essentially these cells have both the mutant and the wild type mitochondria, except where something like this happens and you get sorting of all of the bad ones, or the ones with the mutation, into one of the cells. This would be extremely rare. You would expect that one of the daughter cells would get all one kind, or one mitochondrial genotype, but the other one would kind of have a mix. In either case, you end up with a cell that has all the defective mitochondria in it. And then from there on, you can't recover unless you have a back mutation, and all those cells will suffer there. So mitochondrial mutations are responsible for some not so nice diseases that sap people's energy. So mitochondria are required for energy production. If you get a mutation that completely wipes out mitochondrial function, pretty much you're dead. So I would expect a lot of these mutations would not be like completely cutting off cellular respiration and or electron transport taking place there. And then if you get this in an egg cell passing on to all its future progeny, they would all have that disease. In a lot of cases, mitochondrial diseases occur because some of the cells in the body end up like this, but because the mutation happened after fertilization took place and the organism started growing, you would end up with not with death, but just with a somewhat less severe phenotype. If you're interested in mitochondrial genetics or anything that I'm talking about here, you should see Dr. C in the chemistry department. He's actually a geneticist. Well, he's a biochemist, but he does the genetics of mitochondria as his research specialty. So he knows all about this stuff, and if you're interested, he has research projects and openings for students to work with him. Okay, so fine. This is just a picture, but what does it look like in a real organism? trying my very best. I couldn't get 4 C's for alliterative purposes, but I got 3 right there. So Carl Corren was a scientist who worked with the 4 o'clock plant, and this demonstrated what happens with cytoplasmic inheritance, and what I've said previously, which is that it's entirely from the female plant. So here's the female plant, here's the male plant, and there are 3 different phenotypes that we're going to show here. So these are mutations in chloroplasts, not in mitochondria. Chloroplasts have chlorophyll, which is responsible for harvesting light energy from the sun, and it is a green pigment. So that means that it absorbs light that is what colors? Red and blue. So remember from Principles of Biology 1, right? Because it is green, it does not absorb light in the green spectrum. It's reflecting green light out towards you, so it is absorbing light in the red and blue spectra respectively. All right, so when you don't have chlorophyll or have very little, you have a white color in the leaves. Complete is green, and then in cases like this, you have what's called variegated. These are the very nice plants that you find with speckling on them in the garden section of Lowe's and Home Depot and Walmart and places like that. So in these cases, some of the cells have lost. Let's pretend these are chloroplasts now, and this one isn't making the pigment, or is making the white chlorophyll instead of green. It doesn't work as well. So any cell that loses excuse me, any cell that loses all the good chloroplast genes will make a little patch here. So this is similar to the mosaicism seen in the calico and tortoiseshell cats, where you have random X chromosome inactivation, but here you have random cells that have chloroplasts that have all the chloroplasts are defective for green chlorophyll production, or green enough that you can see it. And then every one of these makes a little patch there. All right, so on this plant, you can see there's some green leaves down below there, but the leaves at the axillary stem where the flowers are coming off are completely white. That means that the reproductive parts and the gametes here will have only the defective chloroplasts in. Here it's green, means that it will have only good chloroplasts, no mutation, and here you get variegation. So it's a random shot. Some of the gametes will have chloroplasts that have good genes in them, and some of them will have the mutation, whatever it is, that makes the pigment turn white instead of green. Same thing for the male plant. Let's start here at the top. So here you can see that all the progeny are completely white-leaved. So even when you have males that have no ability to pass on chloroplasts with the white mutation, everything is white like this. So it is entirely the chloroplasts from the female plant that are giving that phenotype. Here, green, no matter what, even when you have only white chloroplasts containing male gametes. And then the cool thing down here is where you have the variegated, some of the cells will have only chloroplasts with the mutation, and so they will be white. Some of the cells or gametes will have chloroplasts with only the wild type genotype, and they will be green. And then you will have some that have a mixture. So some chloroplasts have the mutation, some chloroplasts are wild type, and then when you get sorting of cells, some of the cells will end up having no good chloroplasts in them, and those are going to end up being the white patches. So as soon as you get to that point, you're stuck here. Now most of the green and the variegated plants are very likely cells that have some chloroplasts that are wild type and some with the mutation, but they have enough with the wild type gene that they make enough chlorophyll that everything is fine. All right, so again, this one is different from everything we've talked about before because the chromosome that we're describing is actually within the chloroplast or the mitochondrion. Everything else is what we call the nuclear genome, or the genome that we've already talked about. So the chromosomes in the nucleus of the cell. Okay, so now we are going to get into another part of genetics called epigenetics, in which you can get heritable genetic change that is not the result of a DNA base pair sequence change. And the example we're going to use is genetic maternal. Whoops, that's the next one. Genetic maternal effect is regular inheritance. Sorry, I'm not thinking right. So genetic maternal effect is where what matters for your phenotype is your mother's genotype, not yours. We're going to get to, I saw genetic maternal effect and I thought my brain went immediately to genomic imprinting, which is the next topic that's coming up. All right, so take this and burn it into your eyelids. Remember that for here. So here's the picture from the front. We talked about the snail. If you look at this snail going from bottom to top, you get a left-hand rotation, so it's going around in this direction, going from bottom to top. In this one, you get a right-hand rotation of the shell, so it's going this direction if you're headed from the bottom to the top. And this is subject to what we're calling genetic maternal effect. So you don't have to worry about figuring, yeah. All right, so we're going to do this for DNA, for when we talk about DNA, but anytime you get a helix, if you want to be able to tell if it's a right-handed or left-handed helix, we're going to use what I've now been forced to call the Facebook test. I used to call it the Fonz test. Who knows who the Fonz is? Put your hand up if you know who the Fonz is. See, just a few, right? What the Fonz always do, what's his signature move that he always does, right? Yeah, he says. Anyway, so, but now it's the Facebook test because everybody knows that this is from Facebook, right? It's sad that I have to move up to the modern era. So if you want to do this, let's look at this one first. So everybody stick out your right hand, just put it in front of you and give the thumbs up sign, right? Now, if you look at the way your thumb is pointing, that's from the bottom to the top, like up in the air. The direction of rotation for a right-handed coil is going to be from your palm to your fingertips. So you can see it's going in this direction, right? Yeah? And that matches this one. So do the same thing with your left hand. Put your left hand out and give the thumbs up. And so going in the direction your thumb is pointing, so from the bottom to the top, now your fingers from your palm are going, now they're going this way. Your thumb is going the wrong, yeah. The way from your knuckles, from your palm to your fingertips. So right hand is going this way, left hand is going this way. And you can see they match there. Now you'll always be able to tell which way a helix is going. And you're ahead of the game for chapter 10. All right. So, genetic maternal effect is caused by the genotype of the female. Remember, I said the egg's got a lot of stuff in it. It's like the Winnebago. It's got food to start with. It's got a map in there. It's got some instructions for how to start building the organism. It's not immediate that genes expression starts once fertilization occurs. So some of the early developmental decisions are made based on either RNA molecules or proteins that are loaded into the oocyte or the egg prior to fertilization, and that's what we've got going here. So the direction of shell coiling is determined by materials that are already in the egg when fertilization happens. Therefore, it's the mother's genotype that matters, not the actual snail we're talking about's genotype. So dextral is a right-handed coil. S+ is dextral, It's dominant to sinistral, which is the left-handed coil, which is, I still object, I'm left-handed, I don't like being, you know, sinister is a bad thing, and that's actually where it came from. So, you know, left-handed people are sinister, so I'm not kidding. So left-handed coil is little s. It's recessive. So we have here a dextral shell snail that is S+, and a sinistral that is SS. After fertilization, the F1 generation is all S+S, right? That makes sense. And here's where the problem comes in. Because normally what you would expect is they would all be dextral because you have an S+ allele which is completely dominant. But the phenotype of the offspring is determined by the genotype of the female parent. The genotype of the female parent was SS, and so it is sinistral. However, the genotype is still the same and the inheritance is still the same. So now we're going to go down here. This particular snail is allowed to self-fertilize. So it's just a monohybrid cross, and you're getting a 1 to 2 to 1 S+ homozygote, S+S heterozygote, and SS homozygote, right? But they are all dextral, even though these two should be dextral, and this one should be sinistral. It should be a 3-to-1 phenotype ratio. But again, it's due to the genotype of the female parent, which in this case was the same one. I wish they would just show it as this snail mated with another male that was the same genotype, make it easier, but you know, that's the female and the male parent there. All right, so what's going to happen in the F3 progeny? So if this one, obviously all the progeny are going to be S+, it's going to be dextral because that's what we got here. So what are the progeny of this one going to be with respect to genotype? If it self-fertilizes, it's only got one allele, right? Little s. So the progeny are all going to be what? Genotype. Little s, right? So what phenotype are they going to be? Genotype is little s. What phenotype are they going to be? They should be sinistral, right? If you're thinking normal things, but, and they actually, yeah, you're right, never mind. It turns out that that's right, that's correct. It's this one that's weird. So this one is going to be then a 1 to 2 to 1 genotype ratio like this, and what are the progeny of this one going to be phenotypically? They're going to all be dextral. That's it. So that's all you have to remember. It's just, and I will tell you when genetic maternal effect is taking place in a problem, so you don't, so you don't have to figure it out. And then all you have to do is look at the genotype of the female parent to determine the phenotype of the offspring. All right, now it's genomic imprinting, which is what my brain told me I was talking about just a minute ago. So this one is an example of epigenetics, where it's not a change in the allele or DNA, but it's marks put upon the DNA during development. So genomic imprinting is when you get an allele from mom and an allele from dad, but they are expressed differently even though they have the exact same phenotype. So the imprinting in this case is methylation marks that's going to be applied to the DNA. The change of the base sequence, so I know we haven't gotten to the molecular part, but the A's, the T's, the G's, and the C's, that has not changed. You will get the same allele from mom as you do from dad, but one of them will be marked for expression and one of them will be marked to be turned off. Because the marks are different if they come from the male and the female parent, during gametogenesis, they will be entirely reprogrammed. So as we'll see, you're going to get one marking from your father's allele and one marking from your mother's allele, but in your gametes, the markings are going to be reset, and we'll see exactly what that is here. So immunoglobulin like growth factor 2 is an example here. So this stimulates fetal growth, and it is one of the classic battle of the sexes that occurs with the male and female inheritance from the genome. So the more IGF, the greater the fetal growth. So males would like, I mean, you know, your sperm aren't sentient or anything, but you would like for your offspring to be as big as possible, because that has a better chance of being, you know, healthier and moving your genes on to future generations, because you'll be better in competitions and so forth. But the male, as the females in the crowd are all going, hey, what the heck, right? Doesn't have to incubate the fetus, right? Gestation is not a male problem. The female's like, hey, you know, too big of babies, especially in species where multiple births are the norm, is not going to be good, because it, for lack of a better word, wears out the female reproductive capacity. And if the offspring are too big, then what happens is you can actually lead to problems with death during delivery of the babies and so forth. And the female wants to be able to reproduce over and over and over again. The male deposits the sperm and then he's gone, but the female got one reproductive tract and has to make it last. So the IGF-2 locusts, the same place here, exactly the same allele, but the maternal allele is imprinted so that it is turned off. So in all, whether you are male or female, the allele you got from your mom is not turned on. The allele you got from dad is turned on. But when you go to do gametogenesis, you want to reprogram that. So the females want to give the allele that's turned off. So in your germline, you reprogram the marks so that instead of having one marked for being on and one allele marked for being off, they're both marked for being off. It's a reset. So every egg gets an IGF-2 allele that is marked for being turned off. And males, somatically, just like females, will have in each cell one allele on and one allele off. But in our sperm, or the spermatogenic pathway, this imprint is removed, it's reset, so that in sperm every single IGF-2 allele is on. So somatically, males and females are exactly the same. But in the germline, it's reset so that you're all either the female turned off or the male turned on. Good, good? All right. Sometimes when you get a mutation, the gene is made completely non-functional. The gene that we're working on in lab that I'm going to have you map is completely broken by the mutation that is in it. So worms with this mutation make sperm that look good and meet the oocyte like they normally should, but fertilization doesn't happen. Because whatever this protein does, which I don't know yet, it makes it so that fertilization can't happen. But many of the alleles that I have in my lab have a DNA base change that makes an amino acid change in the resultant protein that doesn't completely break or make non-functional the protein. It just sort of weakens how the protein can fold and hold together. And that means that under certain conditions, the worms are fine, and under other certain conditions they're sterile, and that is temperature sensitive. So these are conditional alleles. If I grow, so normal growth condition, we grow the worms in lab at 14 degrees, but that's just to make them grow as slowly as possible, to slow down that chemical reaction of life so that they'll last a whole week for you to come back. Normal growth temperature is 20 degrees Celsius, which is about 68 degrees Fahrenheit. So these temperature sensitive alleles at 16 degrees, which is about 60 Fahrenheit, are perfectly fertile. The hermaphrodites make plenty of babies, and you would never know that anything is wrong. But if you take one of these hermaphrodites and move it up to 25 degrees, because of that higher temperature, whatever the protein's function is, it can't do it anymore. We presume that's because when the protein is made, it doesn't fold right, or when it's made, it folds right, but the higher temperature can unfold it, depending on the case. And so worms raised at 25 degrees are completely sterile. At 16 degrees are completely fertile, which makes it really good for us in the lab because we can grow these mutants as a homozygote, whereas the mutant you guys are going to use in lab today, we have to take special measures to keep it going, because as a homozygote it is 100% sterile at any temperature. So it is a non-conditional allele, but conditional alleles, depending on different temperature, will have different phenotypes. What I've talked about is a heat-sensitive allele. So at the lower temperature, which is the permissive temperature, permissive because it allows the protein to function correctly, everything is fine. But at the higher restrictive temperature, something happens and the protein doesn't fold right, and nothing works. There are also cold sensitive alleles, which is just the opposite, where at a normal warmer temperature they function just fine, but at a lower temperature bad things happen and the mutation shows. I also want to talk a little bit about temperature sensitive period. So for the C. elegans mutants, the temperature sensitive period is only during the L4 larval stage. We've talked about that in lab. L4 is the last larval stage before the worms become an adult. That is also the stage at which all the sperm get made. Remember, C. elegans is a sequential hermaphrodite. It makes all the sperm during the L4 stage, then it becomes an adult and switches over to making oocytes instead. So if you raise worms with a temperature sensitive sterility or fertility mutation at 16 degrees, you keep them at 16 degrees all the way through the L4 larval stage, all the sperm get made and they're fine. Anytime after L4, when they become an adult, you can crank them up to 25 degrees and they'll still make babies just fine because all the sperm are already made at the lower temperature. Presumably all the proteins folded fine, and 25 degrees in this case isn't enough to unfold the proteins. You can take the same temperature sensitive worms, grow them at the permissive temperature of 16 degrees, L1, L2, L3, move them to 25 degrees, the non-permissive temperature, just for the L4 stage, and then switch them back to 16 degrees for the rest of their life, and they will be completely sterile because they were at the restrictive temperature during the temperature sensitive period, the time that matters. And it makes complete sense when you think about it. The temperature sensitive period is during L4. That's because that's the only time when the sperm are made. So a friend of mine who works at Rutgers in New Jersey works with the same kind of sterility mutations, but in addition to sperm mutations, he also works with egg mutations, like in the oocyte. He also has temperature sensitive mutations. But because eggs are made throughout the entire lifetime of the hermaphrodite, if he has a temperature sensitive mutation that's sterile at high temperature, if you raise it until it's an adult at 25 degrees, they'll be sterile just like mine. But then if you lower it down to 16 degrees, they immediately become fertile. Well, how come that works for his and not for mine? It's because the eggs are continually made. And so as soon as you move it from the high restrictive temperature to the lower permissive temperature, all of a sudden now everything is fine and the protein gets made okay and everybody's happy. All right. Very cool picture of the Himalayan rabbit. I talked to Dr. Danderson, who actually has grows and has pet rabbits, but he does not have one of these, even though they're really cool. So has anybody ever had a Himalayan rabbit? Yep, they look kind of like this, right? Well, because you live in Alabama. So the reason, anybody else had a Siamese cat, the Blue Point Siamese, same thing, like this, dark on the extremities and light on the body. They have a temperature sensitive or a heat sensitive allele for black pigment. The reason why they're white all over is because at higher temperature the black pigment can't be made, but the extremities, like the ears, the nose, and the feet, and I think the little bunny tail, but we can't see it back there, are at a lower temperature because they're not on the core. They're the appendages, and they're at a low enough temperature that you can actually see it. So it's the same genotype everywhere, but it's because this is at a higher temperature and these are at a lower temperature. Reared at 30 degrees, which is like 90 or some, a little over 90 or something, the higher the temperature, you can see it's reduced. Now you're never going to get completely white, because if you put the bunny at a high enough temperature to get it completely high, you would end up with Hassenpfeffer and not and rabbit stew and not a pet bunny. But I'm thinking it's because, well, it's either the genetics that did it, where it's not a strong enough allele, or raising them at a having them at a higher temperature would sort of have this happen as well. And like I said, the same thing happens with the Siamese cat as happens here. All right, we're going to go on to start chapter 7 now. But I want to make sure again that I'm concerned that you guys are getting what you need. So I have office hours right after this. Anybody who's not been doing so as well as you would want, please show up. And I'm going to send some emails out to people inviting them in, but don't wait for that. Show up today. I want to try to talk to some people that are having having difficulty with understanding or whatever, to figure out what you're doing and try to make some adjustments to how you're studying for the class to hopefully make it better. So please come on and see me after this. Okay, so remember, we skipped chapters. We're not skipping chapter 6, but chapters 3 and 4, because it's just a lot of it is the pedigree stuff. I don't have time to cover the rest of chapter 6, which is interesting. So it talks about fetal diagnostic testing. There's also some information in there on teasing out genetic versus environmental effects using twin studies. So if you're interested in that kind of stuff, I would strongly recommend you read that. It's not that I don't think it's important. It's that I have to pick carefully the topics that we go over in class, and it's not as important as some of the other things. Okay, so previously we've talked about monohybrid crosses, where you just have one cross on one chromosome. Dihybrid crosses, where you have two different genes, but they're on different chromosomes, so you get independent assortment, Mendel's law. What happens with one cross on one chromosome doesn't affect the other chromosome, because the way one homologous pair lines up at metaphase of meiosis 1, either this way or this way, doesn't affect the orientation of how any of the other ones line up. Now we're going to find out that in cases where some genes are on the same chromosome, they all move together because they are linked. They're going together on the same raft, I guess you would say, or the same boat. They're on the same chromosome, and they're inherited together. We're going to also find out how, because of recombination, you can have them not be inherited together, and the degree to which they're not inherited together is dependent on how far apart they are on that chromosome. All right, so independent assortment. Let's go back and talk about this again. Independent assortment is because of interchromosomal recombination. So between chromosomes. Recombination just means a shuffling of alleles. I know that you think recombination just means when two sister chromatids exchange genetic material in prophase of meiosis, but recombination is any time Mendel's second law is applying. So here you have your standard dihybrid cross, homozygous dominant, homozygous recessive for both, only one possible gamete. The F1 generation is a heterozygote for both, and then you can get 4 different possible allelic combinations when you get the gametes forming. So we haven't talked about this before, but we're now going to talk about parental or non-recombinant and non-parental or recombinant gametes. What are the parental and what are the recombinant? The parental are simply what goes together in the original one here, the original parent up at the top. So in this case, both dominants go together. The big A and the big B go together, and the little a and the little b go together. So they are the parental or non-recombinant. It could just as easily be big A's and little b's here, and little a's and big b's over there, and in that case the parental would be those, big A and little b going together and little a and big B going together. It's just how they have it set up here. So these gametes are parental or non-recombinant because they are the original combinations, so big A with big B or little a with little b. The recombinant are the ones that don't match the parentals, a big A with a little b or a little b or a little a with a big B. So far today there haven't been any questions. I want to encourage you, keep asking the questions as we go forward when something doesn't make sense. All right, so now we're going to consider what happens if independent assortment that we've been talking about in Chapter 3 and Chapter 5 isn't happening. So in this case, you have a homozygous or true breeding purple flower long pollen plant and a red flower round pollen plant fertilize purple flowers and long pollen. What does that tell you about the dominance of these alleles? Purple flower and long pollen's dominant. All right, so far so good. Then you go and you get the F2 progeny. Now if this was the standard dihybrid cross, what is the normal phenotype ratio that you should get? 9 to 3 to 3 to 1, right? So that could be 9 because there's a lot of them, and these two are the same, fine. But look at this one down here. The one that got both recessive phenotypes is way higher than the two middle ones. So the 9 to 3 to 3 to 1 definitely is not working here because this guy is definitely not lower than those. In fact, it's more than double. What's happened is that these alleles, the allele for flower color and pollen shape, are actually on the same chromosome. That's called being linked. They're linked together. If they were completely linked, you would expect to only get these two on the end because they would they would always be the original or parent phenotypes. But what's happened here is we've had intra-chromosomal recombination. Intra means between. So we've had recombination here that is the classical way you think of recombination with in meiosis. So what happens is that you can get the gametes so that the long and the purple and the round and the red don't always stay together. These two are the parental phenotypes here on the end, because that was the original flowers. These two in the middle are the recombinants, because now you've got a purple which is dominant with a round which is recessive, and a red which is recessive with a long which is a dominant, right? All right, so both the inter, which is Mendel's second law, independent assortment, and intra, which is exchange of information between non-sister chromatids during meiosis, recombination result in 50% recombinant and 50% non-recombinant gametes. So in this case, you have the chromosomes on different chromosomes. So we're talking about independent assortment here. In one case, the big A and the big B go together. In the other case, the little a and the little b go together. It's just like which way they line up. Do both, in this case, both the big A and the big B line up on one side, the others on the other side. In this case, the little ones line up. Right? Easy enough. We've talked about that. Here are the parental, and here are the recombinant chromosomes. Oops, these are all the parental. Now, in this case, they've lined up the other way. So in this case, big A lines up on the left, little a on the right, little b on the left, big B on the right. So you get big A and little b, and little a and big B. These are the recombinants. So 50% this, 50% that. All right. Now let's look at intrachromosomal recombination. So let's look at the bottom down here first. Here are two homologous chromosomes that are lined up in prophase of meiosis 1. These in the red are sister chromatids. These in the blue are sister chromatids. And what we are going to be getting here is recombination between non-sister chromatids. So this part of the blue and this part of the red are going to be exchanged. See that? So it's showing just the B gene here as an example. But every single gene that's in this blue region and this red region will have recombined. And we talked about this a little bit in that when we talked about if recombination does not occur, then segregation of the alleles occurs at meiosis 1. If it does, then segregation of the alleles occurs at meiosis 2. And in this case, you have recombination that includes only the area to the right of where I'm indicating. So in this case, segregation of the big A from the little a is going to occur at meiosis 1, but all the genes in the area that's shown here, including B, segregation of those alleles only occurs at anaphase of meiosis 2 when the sister chromatids separate from one another. And you get 50% non-recombinant here and 50% recombinant gametes there. Crossing over and recombination occurs in prophase of meiosis 1. Then in anaphase of meiosis 1, the homologous chromosomes separate, but these parts here that are different go to the different sister chromatids. Now in the two daughter cells, one on the left, one on the right, you get separation of the sister chromatids, and these two are going to separate out there. So you end up with recombinant gametes in that way. All right, so I've said that recombination due to Mendel's second law, independent assortment, results in 50% recombinant gametes. I've also just said that recombination here, intrachromosomal recombination also results in 50% recombinant gametes. So why is this screwed up? It should be 50% these two non-recombinant and 50% these two recombinant in the middle. The difference is intrachromosomal recombination, or recombination between non-sister chromatids, does result in 50% recombinant gametes, but only when it occurs. You would expect to get, this is going to happen 50% of the time this happens, 50% of the time this happens. However, this recombination event does not occur half the time. It only occurs when you get a random recombination between A and B. It will happen more frequently as the distance between A and B increases. If you can think of, let's say that between here and here is 100 units. We're going to talk about units in just a second, but divide this into 100 pieces. There's a 1 in 100 chance that you'll get a recombination any of these places, right? If these two genes have only 15 units between them, they're going to recombine a lot less frequently than if they had 60 units in between them, because there's more chances, right? The farther apart they have, the greater number of recombination lottery tickets they have bought, so to speak. Although winning the Powerball does not substantially increase buying one ticket or 100. You have to buy, you'd have to buy millions of tickets for that to happen. But here, it actually works. Okay. So, if genes are completely linked, which is a lie and never happens, then you would have no recombination between them. So we're going to pretend now. So we're looking at tomato plants here, and normal colored leaves is dominant to mottled. So mottled is just like the variegation that we've seen previously, but they're calling it mottled in this case. And normal height, tall, is dominant over dwarf, like this. Right? So we're going to start now doing test crosses with a heterozygote plant to be tested and a homozygote tester plant. Just like when we did test crosses earlier, it's really important that the tester plant, the one on the right here, is homozygous for recessive. Because it's got all recessive alleles, the dominant or recessive alleles received from the other parent will definitely show up in the phenotype, and that is how we tell what's going on. Good so far? So, the tester plant is whether we do crosses with 2 genes involved, or later on in this chapter crosses with 3 genes involved, you're always going to get a chromosome that has all recessive alleles. So in this case, because you have these two together, and because I've said there's 100% linkage, the M and the D capital, the dominant alleles, will always go together. That's this. And the recessive alleles will always go together there. Half the progeny will get this one, half the progeny will get this one. There are no recombinant classes with a big M and a little d, or a little m and a big D, because recombination doesn't happen. They're too close together. So the gametes are 100% parental, and so then the recombinant, or the progeny, will be 100% non-recombinant or parental. What do I mean by parental? Well, the parents had normal and tall and mottled and short. Half normal and tall, half mottled and short. You don't get any that have the recombinant phenotypes. Again, this is sort of in the eye of the beholder. The more crosses you do with the more progeny involved, the greater chance that you're going to see this very low level of recombination that occur. There's almost no genes that are too close together that if you do enough crosses, you can you could see recombination between them. It's just that the closer they get, the fewer times it occurs, until they're so close together that it almost never occurs, but not never. All right. If the genes are completely unlinked, which means they're on different chromosomes, then that means you get 50% recombinant and 50% parental gametes. This is just independent assortment again. The M, big M and little m is on one chromosome, and big D and little d is on a second chromosome. You have 2 4-square Punnett squares. You get 1/4 each of these, and because the gametes are all homozygous recessive, you get even levels of progeny. So if there's complete linkage, you get 100% parental phenotypes. If they're unlinked, as shown here, you get 50% parental and 50% recombinant. If they are linked to some extent but not completely, which is everything else in this chapter, you're going to get something between 50-50 and 0. All right? And again, the percentage or the fraction that are non-recombinant versus recombinant is going to depend on how close the genes are to each other on the chromosome they're on. As the genes get closer and closer, the fraction of non-recombinant or parental phenotypes is going to increase, and the fraction of recombinants is going to decrease until, when they're completely linked, the recombinants end up being 0 and these end up being 100%. Okay? All right. I want to point out again, I really like this author. His writing is good and easy to understand while still being real stuff and not watered down. But he uses some older terms. And he always uses like crossing over to talk about recombination. So when he uses crossing over, it's like me saying recombination. Right? For me, crossing over is when here's 2 chromosomes and the alleles get hooked up at meiosis 1, but this is crossing over. Sometimes the alleles go back to normal and recombination doesn't happen, and sometimes they don't. So I'm making that distinction and he's not. Okay, so non-recombinant progeny, or the parental progeny, are always going to make up the largest class. What do I mean by that? Here is the non-recombinant class, the ones that have the phenotype of the parents. Here's the recombinant class, the ones that do not have the phenotype of the parents. This is all new to you guys. But if you think about it, instead of trying to memorize it, think about why, and that will help you to understand it. Why are the non-recombinants going to always be the biggest class? Because if you're completely linked, that's all you would get. They're together and you never get recombination. The more distance between the 2 genes, the greater the recombinant progeny group is going to be, but it can never be more than the non-recombinant, because if they're not linked at all, it would be 50%. So the minimum fraction of recombinant you can get in a cross is 0 if you get complete linkage. The greatest percentage of recombinant you can get is 50% if the 2 genes are completely unlinked. So we're talking here about somewhere between 0 and 50%. All right. So the non-recombinant is always the biggest cross because it's always less likely that you get a recombination between 2 linked genes than it is likely, because they're together. That's the reason. All right, so the way we map genes and figure out distances between genes on chromosomes genetically is by using recombination frequency. Because I've already told you guys that the farther apart 2 genes are, the greater the recombination frequency. We can measure the distance by using recombination frequency as a proxy. And before the era of sequenced genomes, which I know is before you were born, but it's relatively recent historically, that's all geneticists had, was to measure recombination frequency and see what happens. So the recombination frequency here is just the fraction of the total progeny that are recombinant. So if somebody can get out a calculator, or if somebody has a calculator, and help me out here, it's the fraction of the total progeny that are recombinant. So here we have, I can add that up, is 15, right? So 15 plus this is 108. So 108, I'm not good in front of everybody with this. So what 15 plus 108 is what? 123. So the fraction that are recombinant are 15 divided by 123. 12.2%. And so as we'll see, MAP units, or centimorgans, which we're going to talk about, so you don't have to write it down now, the units of recombination are percent recombination. So these two genes, based on the data that I've given you here, are 12.2 MAP units apart. The thing you're going to do to mess this up is you're going to take the total recombinant and divide it by the total non-recombinant, right? It's not that. It's the fraction of the total progeny that are recombinant. So you have to add these all up together and then take the recombinant and divide it by the total, right? All right. So if M and D were not linked, what genotype ratio would you expect for this? So here, because they are linked, you get more of the non-recombinant or parental and fewer of the recombinant. But if these were not linked and you crossed a heterozygote for both to a homozygous tester, and remember the tester always has to be homozygous for everything, what fraction of each of these would you expect? Well, you would expect 50 for non-recombinant and 50 for recombinant. That's correct. And then between each of these, each divided by 2 would be 25% for each. Right? All right. The author of the book also uses antiquated terminology here, and he uses the antiquated terminology in the questions at the end of the chapter, which is why I'm showing them to you. And also, if you're ever taking a standardized test that uses the antiquated terminology, I want you to know what that means. So it used to be geneticists talked about coupling and repulsion. Now we talk about cis and trans. We've already talked about that. It makes sense because you know cis and trans already from chemistry, and we've already talked about that, right? Cis is on the same and trans is on the different. Instead of sides of a double bond here, we're talking about on the same homologous chromosome or on the other one. If two alleles are in cis, they're going to be on the same homologous chromosome. If they are in trans, one will be on one homolog and one will be on the other one. So in this case, we're talking about and I don't know where they get these fun examples, but the Australian blowfly, I think this one is, has differences in, oh, it's not shown here. Yeah, all right. So it's not shown here, but the wild type color of the body is green. As we'll see, the recessive color is purple. Dominant green, recessive is purple. Dominant here, the puparium, the little case that they incubate in during metamorphosis, is brown. Recessive is going to be black for that. Here, the dominant alleles for green body and brown puparium are in cis because they're on the same chromosome. Here, they are in trans. You can say up here, the wild types are in cis and the mutant alleles are in cis. Here, the wild types are in trans and the mutants are in trans. Note that both of these are genetically equivalent. Both of these are heterozygous for both alleles, and will look the same phenotypically as you can see here. But whether the alleles are in cis or in trans affects what happens when these are crossed with each other. This one would be called a cis heterozygote, and this guy down here would be called a trans heterozygote. As we'll see, it's important in figuring out what's going on with both of these. All right, so the genotype frequency of the progeny is going to depend on whether your alleles are in cis or in trans in the heterozygote, if they're linked. So in this case, we're going to start out with the dominant alleles being in cis, the top panel on the last slide. In every case, we're crossing them to a homozygous recessive tester, so purple body and black puparium. So it's only going to give a chromosome here. That means whatever happens with recombination on the heterozygote being tested is what you actually see. The non-recombinant gametes, which should be the biggest class, are going to be the two dominants together and the two mutants together, and the recombinants are going to be one of each. And you actually see that. So you get 40 of each dominants and recessives here, and 10 of each that has one dominant and one recessive. So where did these come from? Whenever you see the progeny right here, train yourself. See, this bottom line's all the same, right? And I'll always put it on the bottom like this too. The bottom line is just this. So you don't have to pay too much, don't be looking at that because that's just from the tester. It's this top line right here that you get from this parent. So this chromosome comes from that parent. Here, this one matches that one, but one of them, the top one, is coming from this parent. So you either get these two, which is exactly what you've got here, or you get these two over here on the right. In order to get the ones on the right, what has to have happened is you get a recombination in this one, in this fly right here, where you get a recombination, so you get a chromosome that has the P and then the B down, the P plus and then the B. And this one will have the P and then the B plus. So if you just like draw an X in between those on your slide and then trace with your finger from left to right, moving where the X goes, you'll see what happens with the recombination event. So everybody sees how you get the recombinant chromosomes there? All right. So the greatest number of progeny show both dominant or both recessives. That's because these are the parents. But in this case, it's opposite. The mechanics and the reasons why are all the same, right? It's just that in this case, you have a trans heterozygote, where here you had a cis heterozygote. So this is a trans heterozygote. So in this case, the non-recombinant alleles will have one dominant and one recessive each, because that's what they have in the parent. Again, the bottom line down here is provided by the tester organism, and that's all it can give. So in this case, you still get 40s and 10s because the distance between P and B is the same, so the recombination frequency is the same. So 80 plus 20 is 100. This one's easy. So these are, you get 20% recombinants in both cases. But now the non-recombinants are over here and the recombinants are over there. They're switched. So a trap you can fall into is assuming that the two dominants together are parental and one dominant and one recessive being together is recombinant, but that is absolutely not the case. It's only the case if they start out together like this. So there's the difference. Here the greatest number of progeny showed one dominant and one recessive, but the parental or non-recombinant versus recombinant is exactly the same. So with the cis heterozygote, the two dominants or two recessives are parental over here, but with the trans heterozygote, it's just switched. the one of each are the non-recombinant, and the two dominants or two recessives together are the ones that are recombinant progeny or non-parental. All right, I'm going to stop there. We're close to the end. See you guys in lab today if you have lab. Anybody who is, anybody who has time after this, I'll be up at my office, and if we need to go somewhere, we can. But I'd really like to have a conversation with people about how class is going and how you guys are doing stuff, and any advice that I can give to you on how to try to help with your studying. And I need to hear what's going on with you in order to figure out what's happening and how we can try to make a change going forward. Uh-oh, look out. Who's got the